Hello students, welcome to Deepika's Bio World. Today we are going to discuss 11th chapter of Ncert, that is Principles and Processes of Biotechnology. Biotechnology is a multidisciplinary subject which makes of both pure biological science such as botany, zoology, biochemistry, microbiology, molecular biology, etc. and non-biological sciences such as maths, physics, chemical engineering, industrial engineering, etc. Biotechnology deals with the techniques of using living organisms, their products, and the processes related to them which are useful to the mankind. European Federation of Biotechnology defines biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Though we have been using some practices used in biotechnology such as fermentation, which is used since time immemorial, we are using the term biotechnology in restricted sense to refer to only the industrial production and processes used in large scale. The term biotechnology was coined by Carl Ehrlich in 1919 and some call him as the father of biotechnology. Now let us see how the concept of genetic engineering developed. Dear students, in first chapter you studied about asexual and sexual reproduction. You studied that asexual reproduction maintains the genetic information which they carry from their parents through mitosis, and sexual reproduction results in variation because of recombination taking place in meiosis, right? So, when farmers practice plant breeding and animal breeding techniques through the traditional practices, the genes, whether they are good or bad, they are passing on to next generation. There is no choice for farmers to select desirable genes. What if a technique were to be available to isolate only the desired genes and pass on to next generation? As answer to this question, concept of genetic engineering developed. Genetic engineering involves formation of a new DNA which we call as recombinant DNA. This recombinant DNA can have two fates. One is either it enters into the host cell but it doesn't get integrated into the DNA of a host cell. Second chance is it gets integrated with the DNA of the host cell and gets multiplied along with the replicating host DNA. For this to happen, there should be some association with the sequence called origin of replication which is present in the host. Once our gene of interest gets integrated with the host DNA, along with the host DNA it multiplies and multiple copies of a desired gene is produced, and this process is called cloning. Now let us see some details about the first recombinant DNA. First recombinant DNA was produced by two scientists called Stanley Cohen and Herbert Boyer in 1972. How did they create it? They isolated plasmid from a bacterium called Salmonella typhimurium. What is a plasmid? Plasmid is an extra chromosomal circular DNA. It is present outside the chromosomal DNA or the genetic material of bacteria, and its number per cell varies. It is not dependent on a genetic material to replicate. That means plasmid can replicate by its own. In this experiment, scientists took two bacteria. One is Salmonella typhimurium, another one is Escherichia coli. From both the bacteria, plasmids were extracted. Salmonella bacterium plasmid has antibiotic resistant gene meant for resistance against tetracycline. This tetracycline resistant gene is used as the foreign gene, and plasmid of E. coli is used as the vector DNA. Using restriction endonuclease called EcoR1, both the plasmids were cut. Once gene was isolated from the plasmid of Salmonella, it was joined to plasmid of E. coli using a DNA ligase enzyme. It is a ligating enzyme. After that, recombinant DNA plasmid containing tetracycline resistant gene was introduced into another E. coli bacteria. That E. coli bacteria once got transformed with the recombinant DNA multiplies and produces clone of recombinant DNA. In this experiment, plasmid of Salmonella typhimurium acted as the foreign DNA and plasmid of E. coli acted as a host DNA or the vector DNA which carried that plasmid from the Salmonella typhimurium. This produced a recombinant DNA containing DNAs of two different bacteria. Scientists named them as PSC101 in the honor of scientist Stanley Cohen. So dear students, in this process of formation of recombinant DNA, 3 steps are involved. First one is isolating the DNA of our interest, what we call as the desirable gene. Second step is join or integrating that DNA with the vector DNA. Vector DNA is the one which carries this foreign DNA. Third one is multiplying that DNA in a host cell. We have to maintain the host cell and we have to allow that host cell to multiply or clone the foreign DNA. This is the third step. Dear students, hope this introduction and history of biotechnology was useful to you. In next video, we will be discussing tools of genetic engineering. Until then, stay tuned. Thank you.